right, good morning. Welcome. It's great to see everybody here in person at AMP again. This has always been one of my favorite conferences each year. So it's really great to be back in person and to see folks live. It's really exciting. Thank you for coming out. We've got a great workshop planned here. We are going to start off with a short introduction to the Purigen's technology using isotachophoresis for extraction of nucleic acids. We'll do a short introduction to the tech and just a little bit of data on just basics of FFPE extraction. And then we'll turn it over to our speaker, Larry Jennings from Northwestern University, to present his work optimizing for FFPE extraction for methylation arrays. So without further ado, we'll turn it over to Greg Gagne, Principal Application Scientist for Purigen. Thanks.
Thank you, Jay. Thank you, everybody, for coming out and joining us this morning, and special thanks to Larry for presenting his. I'm impressed by this classifier. It looks great. Recent publication as well, so hopefully you're free to look up the paper itself. So as Jay mentioned, I'm just going to try and give a quick primer on how our platform is different than what you've been doing for the last 25 years, and hopefully set up Larry's talk for that. So we're a startup based out in East Bay area with fancy neighbors like Roche Diagnostics and 10x Genomics, etc. If you're more on an academic bent, this technology came out of Stanford University from the Juan Santiago lab. His group is still actively publishing applications of the underlying technology, along with our clinical friends that have adopted the platform. So a lot of the background publication corpus is out of the Santiago lab. Basically what we're looking at here is, how do we work with FFPE tissue? We want to kind of get from what it was invented for. I think that might be a laser pointer. There we go. What it was invented for was basically image analysis and old school pathology. A good friend, Manny Rubin, wrote a book about it. It doesn't really mention anything about molecular approaches, but basically how to use a microscope and predict prognostic and predictive information from that tissue. Of course, we've moved into a new era with molecular pathology based around either sequencing or PCR or platforms like nanostrings, where we've actually trying to take advantage of the same tissue, which is a huge, enormous amount of preserved tissues using formaldehyde-based fixatives. They're really not well posed for molecular analysis because they were developed for microscopy. So the effort to sort of switch over has been a struggle. It's just well ensconced. Basically, this is a great resource, and it's we've built out biobanks now, and everybody's sort of doing the retrospective or prospective approach to try and develop molecular signatures, which obviously Larry will tell us about his this morning, where we can actually subtype a patient at the patient level and get closer to precision medicine. So this is the promise, and that's the challenge. However, these clinical trials do take a lot of patients. Some of the deltas are very subtle, and we need that statistical power to get the needle in the haystack. But there's a number of published examples where it's a challenge, and having numbers in the front end doesn't necessarily mean you're going to get the data out of the back end. So these are all sort of published studies of failure rates starting from blocks or that cohort, and how it pushes through to actual success. So this is an opportunity space for Purigen. It's where we want to operate. Our company has really developed and focused on this out of the Santiago lab the past 8 years or so, and we've been commercial for about 3 years now. And really, I'm not going to read the slide to you, but we really want to optimize the value of this very valuable resource of starting with formaldehyde fixed tissue towards molecular pathology, both at research and clinical ends of the spectrum. How is this different than what we've been doing? We being myself and probably most of you that work in the lab for over 20 years. The top panel here is the current approach, where we're going to make some sort of lysate, typically involving chaotropic salts and or organic solvents, bind that to a surface using differential solubility, wash away not only the cellular debris or inhibitors, contaminants, but also the chaotropic salts and organic solvents you use to start with, because they're really bad for the next step where you're going to use some sort of polymerase or enzyme. We don't want those to carry through. So we're going to call this bind, wash, and elute. And this, is well over 99 point something% of the market right now. This approach in different flavors, but it really hasn't changed since sort of ported it over to carboxylated beads, but very little change. It introduces some challenges, obviously, but we've learned to deal with it. And this is really the only approach we've had available to us for over 2 decades. We're going to use a process called isotachophoresis, and it's incredibly simple and incredibly elegant. And again, this is sort of coming out of the research that originated. I have a slide from a different talk where it's 1970 was one of the first publications using isotachophoresis to separate analytes. So basically, this is a fancy Greek word. It says the same, iso, speed, and aphoresis is movement. So this is describing how ions behave in an electric field. And that's really simple underlying physics. For a given species of ion, it has an intrinsic electrophoretic mobility in that field. And so, for example, to give it a name, every chloride ion will move through a given electric field with the exact same velocity. So isotacho. Nucleic acids are an ion. They have a negatively charged phosphate backbone. So they behave like ions in electric fields. The difference is here, the contrast, we don't bind to anything. We don't wash. There's no washing step. Everything is ions moving through solution on a fluidics device. That gives us some advantages. There's no binding biases. There's no washing biases. There's no eluting biases. Nothing. We don't do those steps. We also don't need to add any inhibitors at the beginning because it's a kinder, gentler approach. So we're not putting a bunch of enzymatic inhibitors that we need to try and get rid of later. So it's going to give us advantages with purity and yield and behavior with your downstream assays. And Larry's going to talk about that after I get through these slides. All right. So in the end, we're hoping to generate better data and take advantage of the FFPE resource that we have at our command. This is the canonical cartoon slide. So on the left here is our fluidics device stretched out from left to right. Molecular biologists love to stretch things out that are usually curvy. But basically what we're doing here is using isotachophoresis and taking advantage of the way ions behave. So we have an ion, and this is the work of our R&D department, identifying the perfect ions to use to make this very stable and robust. We have an ion that moves a little bit slower than nucleic acids. We'll call it a trailing ion. And an ion that moves a little bit faster than nucleic acids. And these are all very well behaved. It's an intrinsic electrophoretic mobility. So we can actually model this in a computer and predict how it'll behave. We put the sample in between these two trailing ion and leading ion, and then turn on electric current. And what happens once you turn the current on, the system immediately self-organizes based on the phoretic mobility, the difference in mobility. So think about a road race or driving down the highway, and it basically self-organizes. And the person that's going the exact same speed as you is the one that's annoying you, because they're always in your blind spot or passing you and on their phone, and they're going the same speed as you. That's self-organized. Same thing as this. So the leading ion will go faster. The nucleic acids will actually get focused, what we call focused, into a very high concentration and high purity in between the ion that's moving a little bit faster and the ion that's moving a little bit slower. The vast majority of the other molecules carry no net charge, so they don't even move. They completely ignore the current in the electric field. They just get left behind. The positively charged will move in the opposite direction. And anything does have a negative net charge, it's very unfavorable, so they move very slowly. They don't have a negatively charged phosphate backbone with a really nice charge mass ratio. Okay, so that gives us really the ability to not only concentrate but also purify the nucleic acids away from everything else. Our last step is to transfer that now purified and concentrated band of nucleic acid into 20 millimolar tris. We do a little trick where we turn off one electrode and turn on a new one. Just move that band into tris, and you recover that from the fluidics device. Here's a quick video. This process takes about an hour. We don't want to rush it. We want to get that nice, pure, concentrated band. We've added cyber green here so you can actually see the DNA in this case. Our fluidics devices might be visible in the background with this lighting, but you can see it's actually banding up quite nicely. White is saturated, so if we turn it on its side, there'd be a really high peak there. It ends up in the tris buffer on the left-hand side. This is a 96 well plate footprint, so these wells are on 9 millimeter centers, so you can use multi-channel pipetters, etc. We've overlaid some animation here just to drive home the idea. These ions are moving a little bit faster, a little bit slower. That defines a physical space where the nucleic acids can exist, right? And ultimately ends up in concentration and purification. Okay, we'll just let that, let's move it on. You just collect your purified materials out of that extraction well with a pipetter. It's very straightforward. Personally, I've trained board certified pathologists that have never used a pipetter in the morning. By the end of the day, they're running the system themselves. The result is obviously higher yield. We don't do any washing of stuff down the drain, and really nice high purity. The underlying physics is very simple, how ions behave in an electric field. It's all automated on a robot. We call this the ionic purification system. The width is about a laptop, and it's all self-contained. You don't need any sort of external computer or anything to run it. It's partnered with a fluidics device and all the reagents you need to make ITP happen correctly. Okay, so you really can walk away with what I've just told you. That understanding of isotachophoresis is enough to run the system and be successful in the pipetting skills. We have a number of different solutions. Today we're focusing on our FFPE solution in the context of Larry's work, the DNA kit, but he also uses the complete kit and not so much the RNA so far, but yeah. But they like the complete kit, which gives you 2 extracts back, an RNA extract and a DNA extract, and the DNA kit. So, but we have, today we'll just focus on FFPE, but we have solutions across the board. The workflow is greatly simplified. I'm not going to go through all the steps here. We do not deparaffinize anything. We just melt it, dilute it out with our lysis buffer. Again, it's very gentle. We don't have any chaotropic salts or those downstream inhibitors. We don't have to get rid of them. It really helps with the system. We do use a thermal mixer to reverse the formaldehyde cross-linking. Hope you know that they're thermolabile. We just let that sit for a little while to reverse the cross-links, free up the nucleic acids away from the binding proteins using protease case step. Transfer that into the chip, and then the robot takes over. Once you've got the robot running, it's automated, and you come back when it's done. It works great. Obviously, I've been talking about increased yield. Across the board with our FFP solutions, we're doing really well. One of the neat things is microRNAs. They're very small. They're very unfavored to bind to a substrate, and so we actually don't do any binding. So we don't wash away the microRNAs, and we found some very consistent increases in small RNA concentration. So there's no length of bias. There's no structural biases as well. Obviously, microRNAs have a very structure-based effect with their GC content, hairpins, and things. We believe our extracts are better quality. We don't degrade any of the material while we're running it. It's in solution. There's no binding steps. There's no eluting steps. We're not actually damaging the DNA as we go. So this is a genome tape, screen tape data where smaller is on the bottom, if you're not familiar with this. And we're getting material without any effort whatsoever that's well north of 20 kb just from normal processing. As far as bias, we did a neat experiment where we compared FFPE-derived read depth, comparing our approach and column-based bind, wash, elute approach relative to pristine, undamaged genomic DNA we bought from Coriell, cfDNA if you're familiar with that. And the biases that we expected, I remember the first time I saw this data, I was very skeptical that this was graphed correctly, but these biases that are fully expected, and we all, this is job security for bioinformaticians, just went away. We got basically the same read depth from FFPE damaged nucleic acids as from the pristine store-bought genomic DNA. So this is really helpful in the real world. In the RNA-seq data, we saw the same thing. This is, there's no evidence of bias here compared to a sonication-based approach as well as the old-fashioned bind, wash, and elute market-leading kits. So the right answer here is random. You get a rectangle, and that's what we see. Does it matter in the real world? The answer is absolutely. We did an experiment calling variants. Luckily, we had the very well sequenced cfDNA. We know the right answer versus the tumor that we were studying, and we've done this for several different blocks. We know the right answer at sites that vary with as little as 10 microns worth of thickness of a surgical resection. This happens to be breast resection. We were able to correctly call all of there's 26 variant locations, 100% accuracy. And what we saw here was similar to what I forgot to point out on the tape station data. And the bias is when you see this bias, you start to see a challenge calling single nucleotide variants, which obviously is very important to this audience. You know, some of this instability, if you will, we plateaued right around 10 microns of input, and we really didn't pull the curve out too far. But even at this is 4 times, for 20 microns worth of the same block, we're still seeing challenges with bias and ability to predict the correct answer for variants. As far as the samples, saving samples, which is really one of our battle cries, again, all of these little circles here are saying we couldn't get measurable yield out of those blocks, and the blue bars are our bars, and we were able to, in one instance working with NCI, we were successful basically saving 100% of the blocks they tested in a little study that we did together that had previously been rejected or failed. So again, this really can help with your statistical power, and in some cases move you from failure to success for that study. So that's what I came to tell you. We are, using a completely orthogonal approach. We don't bind, we don't wash, we don't elute. We don't have a bunch of inhibitors that we had at the beginning that we need to get rid of. Our yields are significantly increased. One of the slides I didn't show was amplifiable yield, which is the real answer. Is this stuff friendly to enzymes, not just giving you a readout on a qubit or a nanodrop, but is it actually useful? And we really have a nice advantage there. We're optimized for maximizing the yield from the smallest amount of tissues. Again, very important to this audience. This isn't a mega prep or something like that for manufacturing. We really are focused on the clinically relevant amounts of material that you have to start with. And we had a cool discussion yesterday with folks working with insects at ports, and you might get 1 or 2 legs off of a mosquito. So that's his share of the surge path block, is a couple of legs. So again, really maximizing the utility of the real world samples that you all get your hands on. Bias free, again, a lot of the biases come from how you handle the stuff along the way. This is in solution. We don't introduce biases. The process isn't capable of introducing biases. It's not like we try to control them. It's just not something that can be done when you're moving ions through an electric field. So that's what I came to tell you. I want to again thank Larry for coming. We're going to be exhibiting at 1425. If you want more in-depth details, please come talk to me at the booth. We're going to save all questions till after Larry's talk. We'll have some time hopefully for that. And then, I'll be available to answer all your questions in depth at the booth the next couple of days. Right? So really great, really thankful for Larry to come and join us. He's going to tell us, I don't want to steal too much of his fire, but really very impressive classifier that he's been using in a clinical setting at Northwestern. Thanks, Larry.
All right, thanks everyone for coming. That was pretty cool technology. So this is not so exciting, I don't the data. I have no conflicts of interest. Everything I say is my own opinion and not that of Purigen. I think they want me to say that. So I'm from Northwestern Memorial Hospital. We're the tertiary care hospital for this clinically integrated network of hospitals. I think we're 13 hospitals now in Chicago and the Chicagoland area. And we're rapidly expanding, probably like a lot of healthcare systems. We're trying to increase our catch basin. And our laboratory, molecular genetics and cytogenetics, are to be the labs, the specialty labs for the entire network. And so we're trying to capture all that volume. So anyway, so we have a need, for, not just dealing with small samples, but dealing with a lot of samples. People might be aware of this paper that came out in 2018. It's a really excellent paper. It's the German Cancer Research Group, or in German, DKFZ group. Basically, up until this point, there had been a number of studies where people had looked at methylation signatures for the diagnosis, classification of brain tumors, ependymomas, or medulloblastomas. This paper actually did a very comprehensive approach where they looked at all brain tumors, and they had a very large group brain tumors, and they created a classifier for classifying all brain tumors. And so our goal was specifically to create our own classifier, because this classifier is on a server in Germany and it's not in a CLIA lab. And so we wanted to create a CLIA lab classifier for ourselves. We did use their data to a very large extent, both for training and for initial validation, and I'll show you some of that. The thing I want to point out with regard to this paper is that the recommendation for the input is 250 to 500 nanogram input for the methylation classifier, which is pretty typical for microarrays to have that kind of recommended input, 250 to 500. When you do a validation, normally we go to the freezer and we pull out the best samples we ever had, because we have lots of that DNA, and we run our validation on that. And then when you validate it and you go live, you get prospective samples where people send you smaller and smaller and more pathetic biopsies all the time. And we wanted to avoid that situation where we go live and we're expecting these brain tumors that are, bigger than the block, bigger than the cassette. And then we get these tiny little biopsies and they want us to do something with it. So from the start, when we started the validation, we wanted to be sure that we can get the most yields for a particular sample. And so we started along those routes. But with regard to the methylation, I'm just going to give a few slides for those people that are not familiar with methylation classifier. Methylation, I think, is really cool. Epigenetics is pretty cool, and methylation is a really great way of looking at that. So with regard to epigenetics, the idea is that every one of our cells has the same DNA, same 3 billion base pairs, and yet cells from different tissues and cells from different tumors look and behave differently. So brain tumors and lung and colon and everything else behave differently. And a cell that's born in the liver doesn't, it becomes a liver cell. It doesn't become a brain cell. You don't get a muscle cell inside the liver. So they retain some of that imprinting or some of that epigenetic modification as they become daughter cells. So bottom line is that tissue have an epigenetic signature. And there's different ways to assess epigenetics. DNA methylation is just one of them. The other ones that work in concert with DNA methylation is histone modification, RNA-associated silencing. But we're just going to focus on DNA methylation. Why? Because it's very easy to study and it's more robust, I would say, than RNA-associated silencing or expression as well. So I think it's a wonderful thing to study. And as shown by the German group, it's a very robust target. So as I mentioned, tissue has its normal signature, normal methylation signature. Layered on top of that is a cancer signature. And when cells become cancerous, a couple things happen. They become overall hypomethylated, like globally hypomethylated, meaning that 90% of the methylation occurs in these highly repetitive sequences that occur throughout our genome. And to keep them silenced, like the jumping genes, to keep those jumping genes silent, we have a lot of methylation, and that's where 90% of the methylation occurs. That largely becomes hypomethylated in cancer, so those jumping genes have a chance to jump and chromosomes rearrange, and so we know that. The other 10% occur in these promoter regions of these genes that regulate gene expression, and those tend to become hypermethylated. And so, as mentioned, there's the normal pattern, and then layered on top of that is a cancer pattern. And so the goal of the methylation array is to assess the methylation pattern and associate that with one particular tumor type or another. So with regard to detecting, it's pretty straightforward. With methylation, the most common way to do it is through a bisulfite conversion, where your cytosine, which is unmethylated, gets deaminated through bisulfite conversion. So that's shown at the top. You add the bisulfite, you oxidize, you get rid of the sulfite, and then you have a uracil. So all your unprotected cytosines become uracils. If it's protected with methylation, it remains a cytosine. You can then detect it by, methylation-specific PCR or sequencing or whatever, or methylation array. So methylation array is pretty much three simple steps that you see in all SNP arrays. The only difference is that the probes are designed for the methylation targets for either converted or unconverted cytosine. So you have probes for each, one for converted, one for unconverted. But other than that, it's just a SNP array. So the steps of SNP array is hybridize. Your patient DNA here is shown in blue and is showing hybridization to the probe. And then it's a primer extension. So then next one, extension and staining, and you do the signal amplification. So you have a biotin, for example, and then you have an antibiotin monoclonal antibody, and you do the signal amplification as shown like that. And then it's just simply about scanning and imaging. And so it's just a SNP array. Just only thing the difference is that you have probes targeting the methylated or unmethylated. Okay, the clinical workflow for this, just for those that are interested in this particular test, clinical workflow is basically a 4-day workflow. We would start on a Monday, and we would do denature, bisulfite conversion. That's an overnight step. Next day, clean out the bisulfite, FFPE restoration, which is an important step, by the way. You can't ignore it. We tried to. And then an overnight DNA amplification. This is not a PCR amplification. This is a linear amplification to get a uniform amplification of the genome, right? And DNA fragmentation, precipitation, resuspension, and hybridization, another overnight step. So you overnight Monday, Tuesday, Wednesday. And then on Thursday, we go through the wash, the primer extension, all those things I showed you with regard to the SNP array, primer extension, wash, and scanning, and then a data analysis when we report on Friday. So that's basically our current workflow. Even though there's that overnight amplification on day 2, again, it's just to get a uniform amplification. You still need a large input, as I mentioned, recommended 250 to 500 nanograms input. So we did validate, as Greg mentioned, we did validate, and the validation was really focused about creating our own classifier so that we would not be using a classifier that's in some uncontrolled server in Germany. So this paper came out, I don't know when, I think it came out in print in August, but was out in early release as early as May. And I'm just jumping to the punchline. I'm just jumping to the summary. Basically, we could create, using the same samples that the German group used, as well as our own validation samples, we could create a classifier that is very much comparable to the German classifier. And that's what's shown here. So the area under the curve is determining sensitivity versus 1 minus specificity. And you can see that the German group classifier has an area under the curve, 0.97. And our classifier is 0.966, so very much the same. And the validation samples used to create this area under the curve, rock curve, was the 1,100 samples that they used. And so we can use the same ones that they used as well. Anyway, so what does that give you? When you've created, the classifier works quite well, and it'll return, because the classifier uses a random forest classifier where there's predetermined groups that we're trying to pigeonhole them into. And so it'll return, if that target tumor is in the classifier, then it'll return a result as well as a score or a confidence score, depending on how confidently it matched with regard to that classifier. So this would be a typical report where the brain classifier would show, for example, it's a GBM, IDH wild type, and the score 1.00 is essentially probability. So it's a very high probability, 0.9999 probability that it's a glioblastoma IDH wild type. And a match because it's over 90% confident that that's what it is. The methylation subclass are different subclasses within that tumor. If, again, those were included in the original training classifier, then you can get a subtype as well. So subclass in this would be an RTK1 with a 0.95 score. So again, a match. So that's basically what the classifier does. There's a lot of other information you can get from the classifier. One of them is copy number changes, right? Because again, it's a SNP array. So you can get copy number information, but you don't, unlike the OncoScan shown here, which is our SNP array, you don't get loss of heterozygosity. But outside of that, it's actually very useful for just calling copy number. And this is a very nice result of copy number done by methylation array versus OncoScan. And you can see clearly that there's a gain of 7 and a loss of 10 chromosomes associated with GBM. But you can also see, for example, EGFR amplification is very nicely shown, or CDKN2A, 2B loss is very nicely shown as well. And so you can classify those things as well. Other things you can do, you could target specific positions such as NGMT, you can classify that. LDH1, methylation, you can classify that perhaps, or CDKN2A, 2B methylation, you can classify that as well, as long as you know what targets you want to look for. So that's actually a little bit trickier than you might imagine, because there's many CPG within a CPG island, and which ones you're going to target and which ones are going to correlate to what you're trying to figure out. That's where the real challenge is. With regard to, just make a point, with regard to the NGMT, there was a very nice paper in 2016 in JMD, and they pointed out, they did a very nice correlation, clinical validation of a couple different positions. So it's very easy to target those specifically and validate to those. So that's what we do for that one. But I'm here really to talk about, what I brought up originally is that we needed a lot of DNA to run this classifier, and we were worried about the samples being so minute that we would not be able to make a classification and we'd be wasting tissue. So really what we were trying to do is trying to figure out what's going to be the best method, and we're going to validate from that from the start. So our methods that we had been using up until this point was primarily Covaris was our workhorse, and we had been using AllPrep as our other validated extraction method for both DNA and RNA, for both AllPrep and Covaris. So the first step was to compare Purigen relative to that. And here I'm just looking at DNA. But this is the total yield in DNA of 25 samples from Purigen versus Covaris versus QIAGEN AllPrep. And you can see these are 25 matched samples. So all were extracted by all three methods. And there's quite a bit of variability, but you can see that there's a clear advantage of Purigen with regard to, I should have done statistics, I suppose, but it seems pretty evident that there's a clear advantage with regard to median, mean, and just range with regard to the Purigen versus Covaris. That didn't mean that every single time the Purigen had a higher yield, but obviously the vast majority of time it did. And so Purigen became our method for, we did validate, because we always want to have two methods validated. We did validate Covaris as well, but we only did enough Covaris so that we can say we have equivalence with regard to if we have a yield or an input of this, we have equivalence with Purigen. We did all of our samples with Purigen from then on. So what are we extracting? I think what Greg was saying is that you're not biasing with regard to length. He showed that you're pretty much extracting everything, whether it's a very short fragment or very long fragment, which, it could be a disadvantage if you really want something that's amplifiable and you're getting nothing but short fragments. So you want to know what you're extracting. So this is just, I think, like 46 samples of just Purigen extracted straight after Purigen extraction. And you can see that there's quite a range. At the top, of course, is the high molecular weight stuff. And I don't think you can probably see this, but at the very top is showing the higher, there must be a point error. At this point here is showing 6,000. So that's the higher marker. And so it basically is, full length DNA. It's about as long as you can get on this particular platform, right? And then on the bottom, it's down to 1. So this down here is showing 1. So 1 to 6,000. And for the most part, just looking at it, you can see that there's a lot of high molecular weight DNA at the top, up near the 6,000. But there are some samples, and it might be kind of dim here. Well, it really doesn't transmit very well at all. Turn on the lights? No, I'm kidding. Sorry. I'll point them out. Let's see. So this one's a very high molecular weight one that I'm pointing out with the green. Very clean, doesn't show much noise. I don't know if you can see this one, if this one's visible at all. It really didn't transmit. I don't know why. It looks much better on my screen. But I'm going to show it to you anyway in another way. So this is the high molecular weight. You can see that it's all very high molecular weight. This is the 6,000 mark. shown here. So this is all like very high molecular weight. And this is a 1, so there's very little that's fragmented. I was surprised by this. This is all FFPE. So I was surprised that we get anything that is, I would have predicted they should all be like we would expect, 200 base pairs or multiples of that. But anyway, so most are high integrity, but then there's some that are like this. And so this is one shown. Obviously, there's still some high molecular weight stuff, but there's a lot of stuff that goes almost down to, 1 base pair. So you get a lot of that stuff as well. So, we're capturing everything with this. The other question that we had was, well, how much tissue do we need? And so invariably, people want to send us a test and they say, well, how much tissue do I need to send? And so we've been trying to get this answer for a little while now. And so what we've tried most recently is to digitally scan the slides and try to estimate what's our yield per, area. And so these are 5 micron sections. So we do a number of 5 micron sections. We'll stain the last one by H&E. And then in this case, this is an H&E stained slide that's been scanned using a free software, if anybody's interested, called QPath. If you want to look at it, it's pretty cool. And it's showing the outline of the individual tissues. You see these are fine needle biopsies. And it's showing the outline of the tissues, and it's also showing in red. Each red is a nucleus that's been identified. So we digitally scanned, we quantified the number of nucleotides. And so this is basically the data from that. This is the yield by area. And the yield is in nanograms, shown on the left, and then that's by area in centimeters squared. And these centimeters squared are, again, 5 micron sections. You can see that there's 2 populations. And so on the one population, can you see this thing? No. Can't see that either. Okay, so you can see there's 2 populations. This population here, and it looks like it is quite linear. And then there's another population, which you can imagine is, seemingly linear as well. And so you'd say, well, why are there 2 populations with regard to area? And that's, I think everybody already knows, but you get a lot of samples like this where there might be a lot of area or a lot of quantified area, but there might be very few sample cells. And so here's one where, the cellularity was measured to be quite variable, as well as things like this, where it's just a fluid with very low cellularity like this. So we get samples like this as well in 5 micron sections. So that could explain the 2 populations, right? So if we go, if we go by, instead of by area, if we go by cellularity, maybe as a better measure. So going back to measure again, but this time we measure the cellularity. You can see here, it looks more like one population, right? But it kind of flattens out as you get more and more cells, it just flattens out. And Greg didn't talk about this in his talk, but you can overload the system as well. You can overload it with too much junk, and you can overload it with just too many cells where you just get a flattening, and you're just not, there's no value added in throwing in more cells or throwing in more tissue, right? So let me go back 1 second. So if I just looked at here, like if I said, okay, I'm going to go up to these 3 points here. All right. So I go up to those 3 points there. It's quite linear, right? The yield is quite linear up to that point. So here I have a R squared of 0.94. And I thought this was interesting. I don't know what to make of this, to be honest. But if you look at the slope says that I set the I don't know what to think about that. Maybe we're getting more yield out of it than I can even, hypothesize. So I don't know how to make sense out of that. Anyway, that's what it came out to be. But nevertheless, it was quite linear with R squared of 0.94. What about area? So going back to area, I mentioned before we had 2 populations. What if I just focused on the first half? And so if I focus on the first half, it's even more linear, actually. So if you have by area, you even get a better linearity of 0.9764 for R squared, which is, again, pretty impressive. Using these formulas of both, the slope, right? I can calculate what we need for our, for our particular tests. So comparing, these are our minimum requirements. So I mentioned that methylation array, they recommended 250 to 500. We validated to 100 nanograms. So 100 nanograms we're confident with. And for our other tests, I just mentioned a couple others just for comparisons. Our comprehensive test, we want 50 nanograms as a minimum. For the Oncomine precision assay, we want 20 nanograms as a minimum. And so this is the minimum number of cells that are calculated based on those slopes, right? So 15,000 cells, 7,500, 3,000. And I think what's striking here is really the amount of area is really small. You know what I mean? We're so used to putting in so much tissue, but this is the total millimeters squared, like not just, for 10 sections like this, but like one section at 3.3 millimeters squared, assuming it's cellular, right? Like the ones I showed you, like the first half, should give you enough, should give you 100 nanograms, and which would be enough for that test. So we're used to asking for, well, just give me 10 or 20 unstained slides or something like that, or 10 micron sections. These are 5 micron sections at 3.3 millimeters squared. You can get enough for that assay. Of course, you want more for repeats and other tests. We're not doing just one test. But nevertheless, I went over these numbers several times, even drive poor Erica into the office with me. I'm saying, am I doing this wrong? There must be something wrong here. And anyway, she says I'm right. Anyway, so a couple of things I wanted to point out, just two things. When we started, this is another, learning moment. When we started the validation, we validated it with a overnight digestion, a gentle overnight digestion. Then when we started getting samples and our technologist says, well, we don't need to do that. We can do a rapid digestion. I know there's a rapid digestion protocol, and they switched to a rapid digestion. And I just noticed that it was getting noisy. The results, especially with regard to the copy number calls, were getting noisy just for that week. And I'm like, what happened? What the heck went wrong? And so we figured out that she did a rapid extraction for that week because she was just trying to get the samples out the door, right? She wanted to get them on the next run. And so this is a particular sample that was rapid digested. We followed up, we did the overnight digestion just for comparison to see, can we do a rapid digestion? Turns out you should not, because it is, although it's not much better, it is better. So things that we couldn't see before. So for example, initially, all right, so we initially had a score of 93, 0.93, and then it went to a score of 1.0 with regard to the call. But the thing that I think is even more important, like here's a gain of 7, but the loss of 10 is totally lost on that earlier sample, whereas the loss of 10 then appears. There's also a CDKN2A, 2B loss that was not called before with the rapid digestion. So this is just a learning moment to say, when you do your validation, you got to stick to it. You can't just, create a shortcut and do a rapid digestion. The overnight digestion is much better with regard to hybrid capture technologies. With regard to amplification base, it doesn't seem to make a difference, but for hybrid capture, it does. So my summary is pretty simple. Purigen provided us higher DNA yield than other methods we validated. It's our workhorse now. We have a couple instruments now, and it's really our go-to for all FFPE. The integrity of FFPE is highly variable. I don't think that's because of the extraction. I think we see it because of the extraction. We get to see those things that are low molecular weight, as well as those things that are high molecular weight. We get to see everything. Downstream quality failures are more likely due to, I think, integrity issues rather than yield. We do have failures, but they're usually because they were decalcified inappropriately or perhaps fixed inappropriately. And methylation array, I think, is a very robust assay, and I think everyone should do it. But seemingly small deviations can greatly affect the results. So the overnight digestion is recommended. All right. Any questions? Thanks for listening.
Yeah.